move on to our first topic today, that is protein. Okay, protein is a complex organic molecule made up of elements, carbon, hydrogen, oxygen, and nitrogen. Each protein is made up of polypeptides, and then polypeptides are made up of numbers of monomers called amino acids. Okay, and then a peptide bond can be broken down to hydrolysis as well as it can be formed through a condensation reaction. There are mainly 20 types of amino acid which can be divided into two groups. First one, the essential and non-essential amino acid. There are 9 essential amino acids, which means they cannot be synthesized by our body. We can obtain this amino acid by a healthy diet, whereas there are 11 non-essential amino acids, which means they can be synthesized by the body. Moving on, let's look at the protein structure. There are 4 states of protein structure. First one is the primary structure, which consists of linear sequence of amino acids. The second one is called the secondary structure, which are called polypeptide chain, forming an alpha helix or beta peptide chains held together by hydrogen bond. The tertiary structure is alpha helix or beta peptide chains folded into a 3-dimensional shape polypeptide chains, for example, enzymes and hormones. Lastly, the quaternary chains, which refers to 2 or more tertiary structures of polypeptides combined to form complex protein molecules, for instance, the hemoglobin.
Lipids are a group of hydrophobic molecules that play an important role in our daily life. So the primary function of lipids are long-term energy storage, insulation, hormone precursors, and a part of our cell membrane. There are 4 categories of lipids. They are triglycerides, phospholipids, steroids, and waxes. Although these groups differ in many respects, but they have one common characteristic, which is insoluble in water. So let's take a closer look at triglycerides. So triglycerides are fats and oils. Fats such as butter and lard are saturated fat. They are solid at room temperature and used by animals for insulation and long-term energy storage. So oils such as olive oil and palm oil, they are liquid at room temperature. They are used by plants for long-term energy storage. They also unsaturated fat. To become a triglyceride, 3 separated fatty acids have to bond with glycerol. So they through process dehydration. A fatty acid that has only single carbon-to-carbon bonds, they are also known as saturated fatty acid. Unsaturated fatty acid have 1 to several double bonds. Double bond result in kinks in the fatty acid chain, which affect the melting point of fatty acid. Phospholipids are similar to triglycerides. They contain glycerol and 2 fatty acids. What's different, the phosphate group is attached to the third carbon of glycerol. Phosphate group is a water-loving. One well-known steroid is cholesterol. Cholesterol is used to stabilize the structure of plasma membrane. And the other one well-known steroid is sex hormones, such as testosterone, estrogen, and progesterone. Wax are a final group of lipids. Wax are non-polar and repel to water. They are found in protective coatings on leaves and outer surface of animals.
So what is enzymes? Enzymes are biological catalysts that direct almost all cellular reaction. Our metabolic reaction depends on it. Without enzymes, the reaction will take too long to complete. So the characteristics of enzymes are it catalyzes specific reaction. It is also highly specific. This characteristic will be further explained by Sakinah in mechanism of enzyme action. So next, most enzymes also requires cofactors, which helps the enzyme to function well and undergo a particular reaction. So enzymes reaction can be slowed down or completely stopped by inhibitors, such as mercury. That's how mercury harms us. And lastly, of course, it speeds up biochemical reactions.
point is the protein. This process started from the nucleus, which the nucleus contains DNA, right? So in this nucleus, it contains DNA, which carries the information for the synthesis of the enzyme. Let's say the red point now in the nucleus is DNA, or to be more specific, is mRNA. It then will leave the nucleus and moves to attach itself to the ribosome. You need to remember that the function of ribosome is to synthesize protein. The ribosome then makes protein based on the mRNA. So we need to remember that in the ribosome, the red point comes as mRNA and then it will leave as protein. Alright, okay then, the synthesized protein then are transported through the space between the RER. Okay, transfer through the space between the RER and before it departed from the RER, the proteins will be wrapped in a basket and then they will part off the membrane of RER. Okay, so from this point, after it left the plasma membrane, the red point now is protein. Okay, now back to the topic. The transport basket then fuse the membranes of Golgi apparatus and it will empty their content into the membranous space. Okay, the membranous space is somewhere inside the Golgi apparatus. Okay, the Golgi apparatus. The proteins are then further modified because bear in mind that the function of Golgi apparatus is to modify protein, right? Okay then, the proteins have been modified. It will be wrapped in a secretory basket and back off from the Golgi and travel to the plasma membrane. Lastly, the basket will then fuse with the plasma membrane before releasing the proteins, which are now enzymes, part of the cell. Okay, then that's it. You did it. It's easy, right? So, if there's any extra question that asks you about the production of extracellular enzyme, what you need to do is just to write all the 7 steps again. It's easy, right? So, all the best in your future undertakings. Bye!
Next, we will learn about mechanism of enzyme action. Every enzyme has their own active site and the active site has a specific shape and charges that complement those of the substrate. So, the shape of substrate must fit the enzyme precisely to ensure the reaction occurs. The way they bind together can be explained by lock and key method. The enzyme can be represented as a lock while the substrate can be represented as a key. Firstly, a specific substrate will arrive at the active site of the enzyme molecule. Then, when the substrate bind together to the active site, it will form an enzyme-substrate complex, just like a key fits into a lock. After the reaction complete, the substrate becomes the product and it will leave and depart from the active site. Now, enzyme is free to bind with other substrate for other reaction.
So now, I will talk about the effects of pH value on the activity of enzymes. The activity of enzymes are affected by acidity or alkalinity of the solution. What is the optimum pH? Optimum pH at which the rate of enzymatic reaction is the fastest. Other fun facts, you guys know most intercellular enzymes exhibit optimal activity at pH values 6 to 8. So changes in the pH value can alter the charges on the active sites. Okay, now let us talk about effects of temperature on the enzyme activity. So as the temperature increases, the velocity of an enzyme reaction will also increases up to a maximum and then will be declines. The increases in temperature will causes increases of kinetic energy of the molecules. So you guys can look at the bell-shaped curve on the graph. So we can conclude that the optimum temperature for most enzymes is 40 degrees Celsius, and when reach 50 degrees Celsius, the enzyme reaction will be denatured.
The next factor is the effect of substrate concentration. Based on the graph, at the low substrate concentration, rate of reaction is directly proportional because many active sites are available. so more chances of collision occur and more product will form. Now let's take a look at the point where the graph is touch constant. At this phase, this is the phase where the reaction reach the maximum rate and it will not increase further because all the enzymes are used. While during high concentration, substrate need to compete with each other for the active site because active sites are very limited. They need to wait until the active site finish catalyzing the substrate. Thus, the enzyme concentration becomes the limiting factor. Lastly, enzyme concentration. Reaction will increase as long as no other factor are limiting the rate and as long as substrate available in excess. Based on the first graph, the reaction will increase until the maximum rate is achieved. And at this phase, the concentration of substrate is become limiting factor. But based on the second graph, if the enzyme concentration is double, the substrate that convert into the product also double, but only when the substrate present in excess.
We did it! We did it! We did it! Yeah!
Thank you for watching guys.
Thanks for watching.
Never
did I have to thank you for watching.
Thanks for watching. You're the best.
For free, so why not me? Why not me? Never did.
Stay safe.
Stay home.
Stay home.
Stay home.
Stay safe. Stay home.